Thank you for that introduction, Elena. Before I begin, I'd like to just thank the organizing committee for this wonderful event today, as well as the Lawson family for this event. As Elena has introduced, I'm a trainee in the lab of Dr. Phil Sherman. And I think it's interesting because as I look across the room, I see a lot of trainees just like myself. And most of us probably come from different backgrounds of research, different models, different systems. But I think that if there's one common thread that really unites us this afternoon is that we're all believers of this undeniable fact that the microbiome plays a very important role in human health and disease. And because of this role, there's a lot of interest in microbial therapies. And one of these therapies is the idea of prebiotics. So for the next 20 minutes, I'll explain to you guys briefly about the concept of prebiotics and perhaps get you guys acquainted with the new idea that they're also more than just bug food. So to start, let me define to you what is a prebiotic. The concept of prebiotics has been around for about 20 years since the initial definition by Robert Freud. But over the 20 years, the definition of prebiotic really hasn't changed very much. For example, the most recent definition of prebiotics is it is a non-digestible compound that through its metabolization by the gut microorganisms modulates composition and or activity of the gut microbiota, thus conferring beneficial physiological effect on the host. So this is a mouthful even for me to read right here, but if there's 3 take-home properties that we should take home is 1, is non-digestible, 2, it modulates activity in the gut, and 3, it produces a benefit on the host. So if we take these 3 properties and now ask ourselves, what exactly is a prebiotic? Well, the list of candidates for prebiotic oligosaccharides have been increasing. There's a lot of new candidates that have emerged in research. But the 3 most well-studied groups of compounds are plant-derived fructans, such as inulin and FOS. FOS stands for fructooligosaccharides, and also GOS, which stands for galactooligosaccharides. And these are plant-derived fibers that we can find in a lot of the daily fruits and vegetables that we consume, such as banana, chicory, asparagus, onions, and garlic. And the third group are HMOs, which stands for human milk oligosaccharides. And these are obviously derived from maternal or donor milk. And what I've shown here are some structures of some HMOs. But realize that in reality, there's over 200 different types of HMO structures identified. So what I've shown you here, these 2 structures, are just a small snapshot. But what I think we can appreciate is notice how the structures of HMOs are much more branched and much more complex than the plant-derived fibers, which are just linear chains. So now that we understand what are some examples of prebiotics, the next question is their consumption actually important? And the answer is absolutely. For example, in the first few months of a baby's life, as the baby is developing the microbiome, one of the most important sources of nutrient is the breast milk. And if we break down the macronutrients inside breast milk, we'll find that actually prebiotics in the form of HMOs constitute a large part of it. And these HMOs serve as important fibers and fermentation substrates for the baby to develop a microbiome, which is why if we compare between a breastfed versus a bottle-fed infant, the development of microbiome is quite different between formula versus breastfed infants. So how exactly do bacteria metabolize these prebiotic fibers? Well, humans are actually quite terrible. Humans don't actually express a lot of enzymes that are necessary for degradation of fiber because we don't simply have the expression for them, which means that most of the fiber remains intact, and as they pass the GI tract, they are intact in the colon. FOS, inulin, and HMO, all of them mostly are intact. But unlike humans, bacteria express a lot of these gene clusters that encode for the enzyme necessary to break down fibers. And these are called polysaccharide utilization loci. And just as an example shown here is the utilization loci for the breakdown of non-digestible starch. And notice how there's 3 series of enzymes needed to break down these polysaccharides. You have these ones lining the outer membrane, which can sense the right type of linkage to the fiber. And then you have these importers, which brings the sugars in. And you have these glycoside hydrolases to break down the linkage. This is a very simple system. But notice that it requires 3 enzymes. And as the complexity of the sugar linkage increases, there's more and more demand for more enzymes. So something with more complex linkage like xylan requires about 21 enzymes, whereas here you only require about 3 for non-digestible starch. And a lot of research has already shown that as you play around with the bacterial genome, start to remove certain expression of certain enzymes, you start to also delay the progression of the microbiome in an organism. So now we understand how exactly do bacteria and why they're good at metabolizing fibers and we're not. But because of this, there's an effect called the two-tier specificity. And this idea is that it's specific on both the fiber that you're giving to the bacteria, and it's also specific to the bacteria that you're using to metabolize it. So in this elegant study done by Seth Rakov down at Harvard, what he did was he took several closely related members of the Bacteroides genus. And what he did was he grew them on these inulin-enriched media, shown here. On the x-axis is measuring OD, which is a measure of the growth of these bacteria. And what he found was that using these closely related members, they actually grow quite differently for these bacteria. This shows that different bacteria metabolize fibers differently. But once he switched the inulin prebiotic to something else like xylem or leaven, he also noticed the growth curves are now different also, which means that if something's an inulin metabolizer, it doesn't mean it's a xylem metabolizer. So therefore, it's specific both to the bacteria and the fiber that it's using. So what do all of this mean? What do they mean in terms of phenotypes of an organism, in terms of health and disease? Well, a lot of these research have been shown in animal studies that they're actually quite profound and very meaningful in terms of health and disease. For example, these are two recent papers published in Cell just a few months ago. In the first paper, this was done by a group in Luxembourg, in which they looked at a group of mice and fed them with fiber diets. One group is fiber-rich diet, shown in green, and the other group is fiber-deprived diet, shown in red. And somewhere in between, he kind of changed the frequency and the type of fiber that they were taking. And once he sequenced these samples, what they found was that they actually diverged quite significantly based on a diet. The fiber-rich diet is very far to the right, and the fiber-free diet is to the left. And what's even more interesting was that when they looked more closer at these microbiome samples, they found the fiber-rich diet had a lot more bacteria that were specialized in fiber breakdown, whereas the fiber-free diet had a lot more bacteria in mucus breakdown. So does this mean that it actually changed the phenotype of the mucus layer? And certainly. Once they took immunofluorescence using a MUC2 antibody to stain for the thickness of the mucus layer in the colon, they found the fiber-rich diet had a lot thicker mucus, while the fiber-free diet had a thinner mucus, suggesting to us that the intestinal epithelial barrier is disrupted purely by deprivation of fiber. So another study also showed a similar story in which they took piglets and now fed them with sialylated milk oligosaccharides. And here they're looking at transcriptomics of different bacterial genes. And what they found was a lot of genes were actually enhanced by the feeding of sialylated milk oligosaccharides. I didn't show you the color coding here of what they actually mean, but these all relate to energy expenditure and growth. And as you would imagine, once they measure the weight of the animals fed with these prebiotics, they also enhance and they have better bone growth. So both of these studies suggest that, at least in animal setting, in both mice and piglets, the feeding of prebiotics to these animals definitely altered the microbiome, and they changed it in a way that augmented their phenotype, improved their improvements, and improved their health. So right now, it shows that the piglets and in mice should care about prebiotics, but what about in humans? So this is a systematic review of 11 RCTs of babies, healthy babies that are taking prebiotic supplementation during their first few months of life. And what they found that, just like in animal studies, they also increase in the abundance of bifidobacteria and lactobacilli. And they've also been shown to reduce stool pH and causing the stool to be softer, and also slightly increase the weight of these babies. But in these studies, clinical impact was not assessed. And this is a growing theme in human studies, is that not a lot of clinical outcomes is known about the effect of prebiotics. But as recent studies have started to come out, there is some impact on clinical outcomes. Just to, for example, this is a study that literally just came out about 2 weeks ago, published in PNAS, in which they looked at lactose intolerance. So they wanted to know, why do some people are lactose intolerant, some are not, and whether prebiotic feeding can have any impact on it. So they took 60 lactose intolerant individuals, and then they fed them with the prebiotic galacto-oligosaccharides for about a month, and they sequenced their microbiome samples. So shown here in this PCOA plot shows you the progression of their microbiome throughout the study. Shown in white, or shown in yellow actually, that's the baseline. That's what they started out with. Shown in red is halfway, midway through the study. And shown in blue is what happens to their microbiome towards the end of the study after 1 month of GOS intake. And what they found was that these individuals had a lot more abundance of bacteria that are related to lactose breakdown. And also in the self-questionnaires, they reported to have less bloating, less flatulence, and less diarrhea. suggesting to you that there is a link between lactose tolerance and a microbiome and how prebiotics can effectively change that. But so far, these studies just shows you that giving certain types of fiber can change the microbiome in a way and improve the individuals. But sometimes it's not entirely about what you're feeding in terms of prebiotics, but it's also about who you're feeding the prebiotic to. In other words, what is the existing microbiome that's taking in the fiber? For example, this is a study that was done recently, published in Cell also recently, in which they wanted to look at the microbiome samples of developing babies in 3 countries. And these 3 countries were Finland, Russia, and Estonia. So geographically, this is a Baltic triangle. But clinically, what's interesting is that these 3 countries, despite being close neighbors, they actually have different incidence of autoimmune diseases. The Russian population has a lower incidence of autoimmune diseases, while Estonia and Finland have higher rates. So they wanted to see whether there was a microbiome involvement in these populations. So when they sequenced the microbiome of these babies, what they found was that absolutely the microbiome diverged based on location. The Russian babies were far to the right. Finland and Estonia kind of mixed between, very similar to each other. When they looked closer, they found out the Russian microbiome, the babies had more bifidobacteria in the resident microbiome, while the Finland and Estonian babies had more Bacteroides. But when they looked at the composition of the HMOs, or the fibers secreted by their mothers, they were actually quite similar. So the types of fiber the babies are eating for the first few months of life are actually quite similar. But because of the difference in the resident microbiome, the type of metabolite produced by these bugs is different. Specifically, the type of LPS produced by these 2 types of prebiotics, or 2 types of microbiome, were different subtypes. The Russian subtype decreased autoimmune activity, while the one from Estonia and Finland actually were able to increase in the autoimmune activity. So this just goes to show that sometimes it's not about what fiber we're feeding to these babies or organisms, but also what kind of microbiome you're feeding it to. So prebiotic structures are also, they're made up of sugar linkages. So a lot of times they also mimic the glycan on top of intestinal epithelial cells. So sometimes they can also act as anti-adhesives to prevent bacterial attachment, as shown here. This is a study done in 2006 in which they added gauze onto cells. And notice how in gauze-added cells, the attachment of the pathogen EHEC is diminished. Just to show there's also, besides effect on microbiome, there's also effect on adhesion of bacteria. So another idea I want to throw out to you guys today is the idea of prebiotic acting as direct immune modulators. This was a study done in 2004 by Dr. Thomas Aweger, who's a clinician scientist currently at SickKids, in which he took T cells from pediatric patients and he exposed them to human milk oligosaccharides. And this is all in an in vitro setting without the presence of a microbiome. And when he did this, what he found was that it actually increased expression of interferon gamma in these cells. And similarly, in our lab, Kathleen Johnson-Henry, who's a technician in the lab, she also performed a similar study, but instead of using lymphocytes, she used intestinal epithelial cells. And like Dr. Thomas Aweger, in an in vitro setting, she exposed these cells to different types of prebiotics. And shown in these shaded bars, there was an increase in IL-10 expression, as well as interferon gamma and other pro-inflammatory cytokines and chemokines. So also, Dr. David Newberg from MIT also took a bunch of colostrum milk oligosaccharides, and what he did was he exposed them to neonatal cells. And he conducted a microarray expression to see the expression of certain cytokines and chemokines, and once again, there was a huge increase in a lot of cytokines and chemokines. So the commonality between all these assays is that it's independent of microbiome, independent of microbiota. He saw there was an induction of pro-inflammatory cytokines and chemokines. So this begs the question is that besides acting on the microbiome, how do prebiotics directly affect the host intestinal epithelial cells? In other words, do they have any direct effects on the host? And that's really the premise of my PhD thesis. So the aim of my project was to assess the ability of prebiotics and human milk oligosaccharides to directly modulate intestinal epithelial barrier function. And to do this, what I did was I cultured intestinal epithelial cell line KGO2s on transwells. After they grew to confluence, I exposed them to 2 types of prebiotic oligosaccharides, inulin and SC-FOS. After 19 hours, I infect them with a bacterial pathogen. And the pathogen I chose is EHEC, which induces a barrier disruption on cell monolayer, and it also induces inflammation of these cells. After 5 hours infection, I check for various measures of barrier function, such as transepithelial electrical resistance, dextran flux, and I also use Westerns and qPCR to look at the underlying cell signaling pathways. So the first result I'll show you is the results on TEER. So TEER stands for transepithelial electrical resistance. So this is a measure of how strong the monolayer is. The higher the value, the better. And when cells are disrupted, when the monolayer barrier is disrupted, the cell's TEER value go down. So the take-home message in this graph is the 3 bars on the right. Shown in black, that's the EHEC control. Notice in 5 hours of infection, the EHEC reduced the TEER to about 25% of control. But in cells that are pre-treated with either inulin or SE-phos, shown in green and blue, is maintained at about 50%. To complement this assay, I also did dextran flux. The way this assay works is you put a fluorescinated dye on top of the cells, and you see how much leakage there is to the bottom. And this graph really is just the reverse of the graph you see in panel A. In black shown is the EHEC control. With a 5-hour infection, there was a huge increase in the amount of dextran flux through the monolayer. But in cells pre-treated with either inulin or SE-phos, either prebiotic reduced the amount of dextran flux, suggesting to you that there's a protection on the barrier integrity. But these are just quantitative assays. What about a qualitative assessment in terms of tight junction localization? So using a Z01 antibody, we stained for the tight junction Z01 shown in green. And notice in control cells, they're very continuous and circumferential. But in cells treated with the bacterial pathogen EHEC, they're now reduced to these punctate spots. And that's indicative of the damage induced by EHEC. But cells that were pre-treated with either prebiotic, inulin, or SE-phos completely just prevented the EHEC-induced barrier disruption, suggesting that there is a benefit on the barrier of these cells. So next, I want to know, well, how are these cells protected? How are these prebiotic exposures able to directly improve the barrier function? So the next thing I did was I selected a panel of tight junctions, and I looked at their expressions. To start, I looked at claudin-1, one of the tight junction proteins expressed in IECs. And what I find is that tight junction claudin-1 was not actually changed by prebiotic exposures. But when I looked at Z01, I do see an improvement. In cells that were challenged by EHEC, inulin and SE-phos treated cells both have higher levels of Z01 protein levels. And similarly, when I looked at qPCR for the expression of their genes, I also noticed that there was an increase in Z01 mRNA levels. And similarly, when we check other tight junctions, such as occludin, we also see a protection on the tight junction expression, suggesting to us that there is a direct effect on the tight junction expressions of intestinal epithelial cells. So how do this come about? What is the signaling pathway? That was my next question. One of the mediators of tight junction expression is the activation of protein kinase C, which is a very promiscuous kinase in the cells that can affect a lot of downstream effects in the cells, such as proliferation, tight junction regulation. So by exposing my cells to inulin and SE-phos in a dose-dependent manner, I see there's a phosphorylation event of PKC at 15 minutes of exposure, which means that if the cells were just growing in a dish, you expose prebiotics at these concentrations, there was a signaling event caused by these direct exposures of prebiotics. And PKC has different types of isoforms. And when I look closer, I found out the specific isoform that was activated by these prebiotics is PKC delta, shown here. But now I have two events. So one is that there's a barrier protection by these prebiotics, and now I have a signaling event of PKC delta. So what is the connection? So using different inhibitors and also siRNA for PKC delta, I lowered the expression of PKC delta and also block its activation using these inhibitors. And what we find is that now the TEER maintenance of these cells is reduced. Once you remove the effect of PKC delta, you no longer have the protection of the barrier function of prebiotics, suggesting to us that the PKC delta works in a way to alter the tight junction expression and protect epithelial barrier function. And that's the summary of the mechanism we have here. So at this point, I found, well, if PKC delta is an activator, is a signaling mediator that's activated by prebiotics, what are other ones? And that's at this point that my committee member, Dr. David Huang, suggested that we should probably take a very widespread global approach to look at all these signaling mediators that's affected by direct exposure to prebiotics. And that's what we did next. So in a collaboration with a group in Saskatchewan, we use phosphoproteomics. So what we have here is a kinome array data. The way a kinome array works is that it has 282 peptides that represent different signaling pathways. And what we're measuring is the phosphorylation status of these kinases. And if there's a change in phosphorylation, it shows you that these pathways are turned on or turned off. So in this raw heat map, these are cells that are exposed to the prebiotics in both unchallenged and challenged state. And right off the bat, you can see there's a lot of difference in this heat map. And a lot of these prebiotics, just by pure exposure, already affected a lot of the phosphorylation status of these cell signaling mediators. In this PCOA plot, we can see that, yes, there's an effect, but between prebiotics, they are actually quite different. For example, ST5 is over here, while inulin is over here. But these just tell you the changes are induced by these prebiotics. The question is, what about function? What kind of cellular function do these cellular changes actually correspond to? So using large databases, we annotated these kinases that were altered, and we find they actually kind of correspond to a widespread of cell signaling pathways and functions. And specifically, a lot of pathways, the canonical pathways are all altered by exposure to these prebiotics. But what was really interesting was that between the prebiotic, for example, between inulin and SC-phos, in the infected state, we find that there's actually quite a bit of difference between two simple prebiotics, which means that, yes, there's a glycan-specific effect on microbiome, but here it shows that there's also a glycan-specific effect in terms of cellular signaling. So we picked now the most annotated pathways, and now I want to see what are the exact specific mediators that are altered by these prebiotics. And when we did this, we found there's a lot of mediators. And just shown here are some of the examples of the pathways that were altered by the exposure to prebiotics. For instance, if we look at NF-kappa B pathway, a lot of mediators such as IRAK4, I-kappa, kappa-alpha were all diminished. And this is just the pathway analysis just to show you the number of mediators that were downregulated by just pure exposure without any microbiome or microbes by the prebiotics. So next we want to see, well, we found these pathways could be changed by exposure to prebiotics. Can we validate these results in vivo using an animal model? So one of the models that we decided to use was the LPS-endotoxemia model. The way this model works was you inject intraperitoneally to these mice, and then you measure the amount of innate immune cytokines like IL-6 in these mice. So in collaboration with Dr. Agostino Piro, who's also at SickKids, we're able to do an animal study like this. At exactly 7 hours, and these are P10 mice, so these are very tiny mice. We give them a single prebiotic gavage with either inulin or SC-phos. And then an hour later, we give them an IP injection of LPS to induce an inflammatory response. And 16 hours later, we sacrifice the mice and check whether there's more or less inflammation after prebiotic intake. When we checked this, we actually found a lot of the pro-inflammatory cytokines and chemokines were reduced after consuming these prebiotics, either inulin or SC-phos. IL-6, IL-17, and Ly6G2, a lot of them were decreased. And what was even more interesting was when we homogenized the colonic tissues and looked at the phosphorylation status that correspond to the pathways we identified, we also see diminished pro-inflammatory signaling mediators. But all of this can say that, well, feeding with prebiotics may be able to induce microbiome change, and therefore I'm seeing these inflammatory effects. So just to check whether microbiome was changed with the ingestion of prebiotics, we also did 16S sequencing. So here showing these bars is the abundance of the major genus inside these mice. And what we see here is there's not much difference. Most of it is all dominated by proteobacteria. There's maybe lower levels in control, but most of the LPS challenged mice all have similar levels of proteobacteria. When we compare the alpha diversity using gen diversity and phylogenetic diversity, we don't see much change between the three groups of mice. And here in the PCOA plot, it shows you that most of the microbiome samples of these mice are clustered closely together and do not diverge based on groups. So all that is to say that most of the inflammatory signaling events we saw through that single intake of prebiotics is most likely dependent on the direct effects on the host tissue as opposed to changes on the microbiome. So with that, I hope I present 3 take-home messages to you. First, prebiotics are promising as microbe-targeted therapeutics. but research from our lab shows that the effects of prebiotics actually extend beyond just microbial substrates or anti-adhesives, that they also have the capacity to directly regulate host cell signaling transduction. And with that, I'd like to thank my lab and as well as the various funding sources. Thank you.